So last time we were discussing about the sources of biomedical signals. We have seen all this type of sources of biomedical signals, and we have seen discuss about the action potential. What do you mean by action potential? Okay, so this phenomena we were discussing, there are some excitable cells. When the cells are excited, the potential is induced, and that can be measured. That is what we have discussed here. Okay. The cell membrane has some semi-permeable characteristics, and because of that, the exchange of sodium and potassium ions takes place, and the action potential is developed. So that is what we have seen. So last time we have discussed some of the terminologies here. Resting potential, action potential we have discussed, depolarization, and what do we mean by repolarization, it was discussed. Sodium pump we have seen. We have discussed all or nothing law. All or nothing law means the cycle of the action potential, it is either fully triggered, the complete cycle happens, or nothing. So that's why it is called as all or nothing law. Then net height of the action potential, this was the height of height of the action potential. That means the actually the amplitude of the voltage, the type of action potential which is developed like this. Okay, the absolute refractory period, relative refractory period, and the propagation rate, these things we'll discuss today. Okay, so this was all discussed last time. Okay. Yeah, I think this was the last point which we were discussing. Now, once the action potential is developed, it is propagated from one nerve cell to the other nerve cell, or this holds true for any other type of cells. Okay, and that is how the signal propagates. And this is the complete cycle of action potential. That we call it as the typical waveform of action potential. Okay, so in the resting state, it has the potential of around minus 70 millivolt, and after it is triggered somewhere here, then the depolarization happens, and some positive potential of around 20 millivolt it is generated. We call it as the action potential. Then after that, again the cell is repolarized and reaches to its original value. Okay, so that is the complete cycle. Now the same thing is explained in this diagram also. Okay, so we have sodium ions indicated by this green color having positive charge, and the potassium ions indicated by these triangles here, okay, which are inside the cell, and the sodium ions are outside the cells. Okay, so during resting state, the inside of the cell, it is at negative potential, which is indicated by this negative signs here, what you see. Okay, whereas the outer side, it is having positive potential. Outside it is the positive potential, and this is the cell membrane. This is the cell membrane, or we can say the boundary. And this C, D, and E, these are the channels. So C is the sodium channel, D is the potassium channel, and this E, this is called as sodium potassium pump. Okay, so the first figure, this one, this is during the resting state, this one. So this is during the resting state, there is around minus 70 millivolt. Okay, now the trigger happens somewhere here the cell is stimulated or excited, okay, then it starts depolarizing. Now during this process, the sodium channel opens. What you see here, the sodium channel opens, and this sodium ions, they transfer from outside to the inside of the cell. Okay, so that means the overall positive charge inside the cell, as you can see here in this diagram, the overall positive charge, it increases, and this is point number, this is here point number 2, that is the depolarization is happening here. This is the process, that is 2. Okay, so the charge is becoming more and more positive, and it reaches to peak here. And after that point, the gates, sodium channels, they are closed. So this sodium channel, it is closed here, and the potassium channels opens. So once the potassium channel opens, then the potassium ions, they are transferred from inside of the cell to the outside of the cell through this potassium channel. And this is actually step number 3. And the potential, action potential, it starts reducing, and that is called as again repolarization. Okay, now after certain point, this sodium channels also close, and this potassium pump opens. This is the sodium potassium pump which opens, and this pump opens the sodium and potassium ions. So for it allows to enter 3 sodium ions inside, indicated by this green color, and 2 potassium ions outside. So these 2 potassium ions are going inside, outside, and 3 sodium ions are entering inside. So after that, the cell again reaches to a resting state, and it is ready for next cycle. So this is what happens during the complete cycle of this action potential. Okay, again this is the same thing. This is the resting state, that means it is polarized, inside is negative. Okay, inside is negative. It is then it is stimulated. After stimulation, the negative charge, it starts decreasing, and once it is fully depolarized, the inside becomes positive, right? And again it is a state of repolarization. That is what we have seen in the previous diagram. Okay, so this is the complete cycle of electrical activity inside it is inside a muscle or nerve cell. Muscle or nerve cell. Okay, now let us see some of the terminologies associated with this electrical activity. This we already discussed, all or nothing law. That means the action potential is always the same for any given cell, irrespective of the method by which cell is excited and intensity of the stimulus. So we have seen earlier, when the cell is excited, one cycle of this action potential happens. Okay. Now the we have seen during resting state, this is minus 70 millivolt, and once it is depolarized, then it is plus 20 millivolt. Now this amplitude 20 millivolt, and during resting state it is minus 70 millivolt, and after trigger it reaches to 20 millivolt. So these amplitude levels are, they do not depend on the stimulus or the amount of excitation, irrespective of the method or the amount of excitation, this amplitude remains same. So that is all or nothing. Net height of the action potential. So that is the difference between the peak of the action potential and the resting potential. So this potential, it is called as net height of the action potential. Okay. Absolute refractory period. So the brief period of time during which the cell cannot respond to any new stimulus. So this is absolute refractory period. So that is somewhere here. Okay. So I think it is shown here. So this is refractory period, what is shown here. So this is the refractory period. So during this time, the cell cannot be triggered again. So new trigger does not cannot stimulate the cell. So that is absolute refractory period. Then relative refractory period. So it is the period of time during which another action potential can be obtained with a much stronger stimulation. So that is relative refractory period. Okay. Then we have seen earlier this action potential, it propagates from one cell or one neuron to the other neuron. Okay. So we have seen so this negative, you can see in this diagram the potential, the negative charge, the positive one inside, it keeps on transmitting from one neuron to other neuron as shown in this diagram. Okay, so this rate, it is called as a propagation rate. So the rate at which an action potential moves down a fiber or propagated from cell to cell is called propagation rate. And for nerves, in case of nervous system, this is around 20 to 140 millisecond meter per second. Sorry. For in case of nerve conduction, it is 20 to 140 meter per second, whereas for heart muscles, this is 0.2 to 0.4 meter per second. Okay, so it's the special time delay fibers between the atria and the ventricles. That is 0.0 to 3 to 0.05 meters per second. So these are the timings with which the potential propagates from one cell to the other cell. Okay, so this completes this part of the presentation. I will load the next presentation. Now next, in the next part, we are going to discuss about the different types of bioelectric signals. That means what is ECG, what is EEG, what is EMG, and so on, ENG, ERG, EOG. So the different types of electric signals that we are going to discuss. So as I said, we are going to discuss about the different bioelectric signals. Okay. Now bioelectric signals, this is nothing but the electrical activity of various cells, which results in some biopotentials. Okay. Now this electrical activity, this is associated with different organs of the body. That means this can be about the heart. So if it is about the heart, then it is called as ECG. Then the electrical activity of the muscles, it is called as EMG, that is electromyograph. Then electrical activity of brain, it is in electroencephalograph, EEG. So there are many like this. So those things we are going to discuss today. Okay. So these are some typical values or the ranges and the types of electrodes used to measure this type of bioelectric signals. So ECG, the frequency range is 0.05 to 120 hertz. 100 hertz, it is the typical value. Okay, and the signal amplitude, it is 0.1 to 5 millivolt. Typical value 1 millivolt. And the type of electrodes used are the skin electrodes for this measurement of ECG. Then EEG, that is electroencephalograph. So electroencephalograph, the frequency range is 0.1 to 100 hertz, and the signal amplitude, it is 2 to 200 microvolts. Typical value 50 microvolt, and the type of electrodes used are the scalp electrodes, so which are attached on the scalp here. Okay. EMG, that is electromyogram related to the muscles. So the frequency range is here somewhat higher. So it is 5 to 3000 hertz. Typical value 100 hertz. Magnitude 0.1 to 5 millivolt. Okay. And the type of electrodes used here are needle electrodes. Then ERG and EOG, these are related to our eyes. Electroretinograph. So the frequency 0 to 20 hertz, and the amplitude 0.5 to 0.5 microvolt to 1 millivolt, and the types of electrodes used are contact electrodes. Then EOG, this is again electrooculograph, 0 to 100 hertz the frequency, amplitude 10 microvolt to 3.5 millivolt, and the electrodes are again contact electrodes. Okay. Now when you see this terminology, the last G, sometimes we use when we say, for example, ECG, then the last alphabet, it is G. So this G stands for gram, a graph. Okay. There are two terminologies here actually. When I say electrocardiograph and sometimes we call it as electrocardiogram. So electrocardiogram, actually from the name itself, it is clear. When we say graph, that means it is some device or the plotting mechanism. We call it as the electrocardiograph. Electrocardiograph. Whereas electrocardiogram, gram means it is the actual signal or the process of doing that. Okay. Now okay, so we'll start with the first one, that is electrocardiograph, that is ECG. Okay, so in this ECG, it is actually the electrical signals associated with working of our heart. Okay, now these are different parts of our heart. Here there is one node called as AVN, atrioventricular node, located at this point. So this is the point where actually the electrical activity starts, and later on the contraction, expansion of the heart, it starts. This is that sinoatrial node. The sinoatrial node, this is the point where the electrical activity starts, not here. Okay. And from that it propagates to different muscles, and the contraction and expansion of different parts of the heart happens, and that is how it works as a circulatory pump. The pumping action happens. Okay, now the details about this, how actually the heart works, how this pumping action takes place, and the associated signals generated because of that. We are going to sorry, we are going to study this in the next unit, that is unit number 2. So at this point, I am not going to discuss the detailed working of our heart, but the purpose here, it is just to introduce you what are the different types of electrical signals. So the first one which we are going to start with, it is ECG, related to the electrical activity of the heart. Okay. Now again, this diagram shows the working of the heart, where the various nodes are there, right? Purkinje fibers. These are actually the fibers where these potentials are generated and transmitted. So that all those things we are going to discuss in the next unit. So right now, I will not discuss here the detailed working of the heart. So this just indicates the electroconduction of our heart muscles. Okay. And this is one complete cycle of the ECG waveform. Okay. When we plot ECG for any normal person, this is how it looks like. And this is the standard pattern. Now, it is divided into 5 parts: P, Q, R, S, and T. Okay. Now, later on, when we'll study unit number 2, at that time, we'll see how exactly one complete cycle of pumping action takes place in the heart, and what are the associated electrical activity. That means what is the meaning of this P wave, what is the meaning of this R wave, which is actually the peak. R is the peak, S is the bottom peak. So what happens at this point P, what happens at point Q, at point R, at point S, point T, and so on. So that we are going to discuss in the unit number 2. Okay, so right now, just to give you idea that ECG signal, it looks like this, and it is divided into this 4 parts: PQ, 5 parts: PQ, RS, T. Okay, this is the actual plot of the ECG signal. Some typical waveform. Now here, again, if you see this, there is a P waveform here, Q, R, S, T, and there is a U also. So this U, it is sometimes this U is visible. Okay. Then after that, again the P wave, that means the start of the next cycle. So this is from this point to this point. This is one complete cycle of the heart, electrical activity of our heart. Okay, the complete heart cycle. Now details about this, as I said, we are going to discuss later. So this is just to give you idea about how actually the ECG looks like. And in our subject, as I told you earlier also, the most important ECG signals are related to related to the heart, that is ECG, and brain. So these two things we are going to study later, and the related signal processing of that. Okay, so that is this ECG. The next one, electroencephalogram, and in short we call it as EEG. And this is related to the brain, electrical activity of our brain. So ECG measures the potential fluctuations recorded from the brain. So the electric potential fluctuations recorded from the brain, that is EEG. Okay. Now, for measurement of this EEG, we have to use some electrodes. Now again, in the later part of this unit, we are going to study different types of electrodes. What are the types of electrodes? What are their applications? And what are the problems associated with these electrodes? That means noise, artifacts, and so on. That we are going to discuss later in the same unit. That is the last point in unit number 1. Okay, maybe next lecture we'll start discussion about the electrodes. Okay, now to measure this EEG signal, which is related to the electrical activity of brain, three types of electrodes are used. Scalp electrodes. These scalp electrodes are like a surface electrodes. Okay, so these are mounted on the surface of the brain, outside on the brain. That's why the scalp. Okay, it is actually okay. Electrode cap, relatively far. It is actually just like a cap which is mounted externally on at various points on the brain. Okay, and as you know, since it is mounted externally, it is relatively far away from the actual brain. Okay, so that is actually the traditional EEG, the regular EEG what we use. Okay, but sometimes the other types of electrodes are used which can give you more information, because scalp, it is measured out from the external, so obviously it is more susceptible to noise. Then next one, it is cortical, so that means the surface area of the brain, outer surface of the brain, that is cortex. Okay, so electrodes are placed on the exposed surface of the brain, that is the outer surface of the brain, called as the cortex. Okay, so those are these type of electrodes are called as cortical electrodes, and this is actually directly touching the brains. These are directly touching the brains. Then the third one, the depth electrodes. Now these depth electrodes are again thin insulated needles. These are the needles which are inserted, placed into the neural tissue of the brain. So again, they are directly touching to the neural tissues of the brain, and these are also called as neural probes. Like your multimeter probes, when you, if you want to measure the voltage using the multimeter, you directly touch the equipment where you want to measure the voltage. So in the same manner, these depth electrodes, these are directly touching the nerve tissues. Okay, but surprisingly, there is a very little damage which is caused because of this type of electrodes. Okay, now these electrodes, they record the fluctuating potentials, and which is a superposition, of the field potentials produced by a variety of active neural current generators within the volume conductor system. Okay, so I will just explain all these things. Now these electrodes here, these are actually different types of electrodes. They will, for example, if this is electrode number 1, electrode number 2, electrode number 3, electrode number 4, and so on. So these different electrodes, they will measure, or they will sense different the potential at different points along the brain, right? And finally, what our equipment displays, it is the superposition of all these principles. That means the addition of all these field potentials produced by variety of active neuronal current generators. These are the actually acting as current generators. So the current generator 1, current generator 2, current generator 3, current generator 4, like that. And finally it is summation of all. So that's why it is called as superposition. Okay, within the volume conductor system. Now this terminology called as the volume conductor, here volume conductor means actually when this you are inserting this electrode, it is touching some part of the brain, but this electrode, it will measure actually the electrical activity which it is not directly touching, or it may be somewhat nearby. Okay, and because of some phenomena called as the volume conduction, this electrical activity of the nearby cells, it is sensed by electrode number 1, like that, or any other electrode. So that is called as volume conductor. That means without actually directly touching the particular cells, this electrical activity can be measured by the phenomena called as volume conduction. That is called as volume conductor medium. Okay, now the recorded potentials from the brain are called as brain waves, and the entire record of this brain waves, it is called as electroencephalogram, that is EEG. So the entire record, it is called as electroencephalogram, and the actual graph what you get, it is called as graph, electroencephalograph. Electroencephalograph and electroencephalogram, these are two different things. Then the brain recordings demonstrate continuous oscillating electric activity. So this type of electrical signals are generated inside the brain, which are measured by the electrodes, as we have discussed earlier, and the intensity and pattern of recording. So intensity and pattern of this signal, what is recorded, it is determined by the overall excitation of the brain. Okay, for example, this is our brain, and you are connecting the electrodes at this point, at this point, at this point. Okay, so actually whatever electrical activity it is measuring, it is the overall pattern of the brain, not only at that particular point. That is the meaning of this. Okay, now this we have discussed in the earlier lecture. The frequencies associated with this EEG are alpha waves, that is 8 to 13 hertz. Now this frequency, it occurs when the subject is awake but rested. Subject means, in this case, the person or the object which is being used for carrying out measurements. So in the medical terminology, it is called as subject. Okay, so when the subject is awake or under steady state, that means taking rest, our brain is taking rest, then the frequency range is 8 to 13 hertz. If we are undergoing with intense mental activity, then in that case, the frequency range is somewhat higher. So that is 14 to 30 hertz. Whereas theta wave, that corresponds to emotional stress or the disappointment, and that range is around 4 to 7 hertz. The frequency is less than 3.5 hertz. This occur when we are in deep sleep, or it may happen in case of certain brain diseases also. Okay, so these are the different types of frequency ranges we get for our EEG signal. This is how a typical EEG, that is electroencephalograph, it looks like. So as we have seen earlier, delta waves, theta, alpha, beta, and gamma waves. Then the next one, electroneurogram. Now, earlier we have discussed about electroencephalograph or gram. Then it was about the brain. Now here, electroneurogram, this is related to a nervous system, our central nervous system. So the central, it measures the electrical activity of neurons in the central nervous system or the peripheral nervous system. So that is called as ENG. Okay, and it is done by placing a electrode in the neural tissue to record neuron action potential or of one or more group of neurons. So a electrode is placed in the neural tissue to measure this ENG, called as electroneurogram, for one or more group of neurons. So this ENG actually it measures the conduction velocity. It measures the conduction velocity in a peripheral nerve. Now we have seen earlier in the previous presentation, there is some negative charge inside the during resting state. There is a negative charge, and outside there is a positive charge. Okay, and after excitation, it happens. It reverses. That means after excitation, the inside becomes positive and the outside becomes negative. Okay, and this propagates, as we have seen earlier in one of the slide during animation. During animation, I have shown you that this charge it propagates from one cell to the other cell or from one neuron to the other neuron. So that is called as conduction velocity in a okay. So this ENG, during this ENG, actually we are measuring the conduction velocity. So how it is done, that we'll discuss in the next slide. Okay. So this is done by stimulating a motor nerve at 2 points. So we stimulate this nerve at 2 points. Let us say, suppose at this is point 1 and another point 2, and we measure the time required for the signal to travel from one point to the other point, and from that we find out the velocity. That is actually this ENG, electroneurogram. So this conduction velocity shows nerves regenerating following some nerve velocity. Okay, now let us see how this is done. So as I told, during this ENG, we are actually carrying out the measurement of conduction velocity. So it is measurement of neural conduction velocity via measurement of latency of evoked electrical response. Latency of evoked electrical response. Evoked electrical response means when you give some stimulation, what response we get after stimulation, that is called as the evoked response. Okay, so we measure its velocity by stimulating this at 2 different points. Okay, so let us see. For example, now here, if we can, as I said, we measure this potential at 2 points. So at this point, you can see there are some electrodes connected. At this point, another set of electrodes is connected. Okay, and here we get this measurement. This is the output of these electrodes at this point, and at this point near the elbow, this is the output of these electrodes. Okay, and the stimulus actually it is given to the finger. Stimulus it is given to the finger, and because of that it is excited, and this action potential it will start transmitting along this direction. Okay, so the time required for this potential to travel this distance, that from that we can calculate its velocity. So this is point S1, this is point S2 for these electrodes. Okay, so we measure this voltages. So for example, this is the waveform at point S1. This is the waveform at point S2. And this is the pulse which is traveling in this direction. Okay. So we measure length L1. We measure this length L2, L1 and L2. So L1 d upon L1 minus L2, that gives us this velocity. So d is the distance between these electrodes. So here this is the d. D is the distance between these electrodes, as you can see here. So d upon L1 minus L2, that gives us the velocity, and with the help of some electrical devices, it can be directly measured. So this is electroneurogram. Okay. The next one, it is electromyogram. Myo means it is related to muscles. Okay. And usually we use surface electrodes for this. So surface electrodes which are connected at the surface of the screen, and those measure these electrical signals generated by our muscles. EMG records from multiple SMU. SMU means single motor unit. So when you say motor unit, it is actually a fiber of our muscle. Fiber of our muscle. So when you insert a micro electrode, it can measure potential electrical activity from a single motor unit. Okay. But when you use some surface electrode, like some plate which is mounting on the surface, for example, this is hand. Okay. So this surface electrode, it will take measurements from all different points. It will sense the electrical activity from various points. So that's why you get it actually the superimposed signal, superimposition of all these signals collectively. That is what we can measure here. Can be used only with superficial muscles. Superficial muscles means which are located on the outer side of the body, immediately nearby the skin. So those are called as superficial muscles. For other types of muscles, that means if the muscles are lying inside or the interior, then in that case you cannot use surface electrodes. Okay. So if you use surface electrodes, it is sensitive to electrical activity over wide area. Insertion electrodes, the electrodes which are inserted directly in the skin, those are called as insertion electrodes. So these insertion electrodes and fine-tipped electrodes can be used to localize the potential recorded from isolated SMU. SMU means single motor unit, as I told you. So if I want to find out the potential related to a single fiber, muscle fiber, that called as SMU, then in that case you can use the fine tipped electrodes. Okay. So this is about this EMG. Now the shape of this single motor unit can indicate peripheral neuropathy or can be modified by some disease. If there is any disease, the shape of the potential output of this SMU, it can be different. Okay, so this is about EMG, that is electromyograph, and this is how it looks like. So this is diagram A, maybe the electrical potential or the EMG from a single motor unit, SMU. This may be from this different fiber or called as the different SMU. And as I said, if you are using a surface electrode, then it will actually give you superposition of all these signals. Okay, so that's why this is actually what you see here. It is the addition of all the signals. So that's why it looks like some random signal. Okay, so that is C. It is showing the individual units can no longer be distinguished. So in this part C, you cannot distinguish any single unit SMU because it is the addition of that. And if there is a strong muscular contraction, then it will show an interference pattern here, as shown in this figure D. Okay. So this is in short about EMG. So this is just an introduction. Actually, these topics are very big topic, and this is one typical graph of how this EMG looks like. Electromyograph looks like after any contraction or expansion of the muscle takes place. Okay, so today we'll stop here, and next time we'll discuss about our eyes, that is the graph related to eyes, ERG, EOG, like that, electroretinograph, electrooculograph, that we'll discuss next time, and that will be end of this topic. Then after that, next time we'll start the electrodes also, study of different types of electrodes used to measure all these different types of signals. So that is for today. If you have any questions to ask, I will answer. Otherwise, we will stop. Any questions?